Swayam Prabha. Digital India. Educated India.
Welcome back to the lecture series in animal physiology. So we are into the week 10 and we have finished the first lecture. We are into the second lecture. So just let me register that out. Week 10th and lecture 2. Okay. So now first of all, let's discuss about one other concept, about where we finished the previous class from there. So in the previous class, we talked about that the impure blood from the upper and the lower part, or rather the superior and the inferior part of the body, comes to the right atrium. From the right atrium, it moves to the right ventricle, and from the right ventricle, it is pumped all the way to the lungs, and from the lungs, the blood gets purified, and the purified blood comes back to the left atrium to the left ventricle, and from the left ventricle, it is pumped all over the body. So just follow me through the picture. So here you have the right atrium where the impure blood is coming. It is moving to the right ventricle. Right ventricle, it is going to the lungs. From the lungs, the pure blood is coming to the left atrium. From the left atrium, it moves to the left ventricle and it is being pumped out. So if we have to break up this problem, you can break this problem as events. What do I mean by that? That means the first event which is happening here is out here. Event 1, the filling of the right atrium, E1, where E stands for event. Okay. Now, the second event which is happening parallelly, simultaneously or with a slight delay, is filling of the left atrium, the upper chamber, the left upper chamber. Okay. So if this one is E1, the first event, then this one is E2, followed by this. The second event which is happening at the lower chambers, where the impure blood is moving to the right ventricle. Okay. And that is your E3 event. Whereas parallelly, there is a fourth event which have started on the left ventricle, which is represented in red color, and I am shading the left ventricle. That is your E4. So E1 followed by E2 followed by E3 followed by E4, and the sequence of events are moving like this. Now, these 4 events which are taking place at 4 different chambers of the heart. Now, first basic concept is, it is like, have you seen those lights which switches on one after another like this, like a wave in a pattern? So there is a pattern, and this pattern is being followed here: E1, E2, E3, E4, E1, E2, E3, E4, E1, E2, E3, E4, and in that process there is something for this event to take place. So now let's go to the second concept in that line. So if we say E1, E2, E3, E4. So, for each event there is a time span. If event 1 in terms of, if we plot event 1, so if this is when the event 1 is happening, okay, I'll have to use another color. So, this is how the event 2 will proceed. Okay. This is how the event 3 will proceed, and this is how, say, event 4 is proceeding. And if I number them like this now, E1, E2, and by the way, I am not using the same color code as I used in the previous slide, so do not get confused. Okay. So, you will observe that when E1 is happening, the same, there is something called a Venn diagram. If you have done set theory, you will realize that something like this. This is one event which is happening, there is an overlapping event which is happening, there is a third overlapping event which is happening, there is a fourth overlapping event which is happening, maybe like this or something like this. So, none of the individual events are following a unique time frame. Within that time frame, another event has started, and maybe by the time these two events has wind up, then the third event has started, or it is also kind of overlapped with some of the time windows. So, that is why when I drew this graph E1, E2, E3, E4, you will be saying there is always a overlapping zones between the different events, and this overlapping pattern. So, there is an unique pattern of this overlapping stuff. So, there is a time span for event 1, there is a specified time in terms of seconds, this many seconds E1 will happen. It means the upper right chamber will be filled, right. Event 2, when the upper left chamber will be filled, what will be the time? Then from the upper right chamber, the fluid will move to E3 event. There is certain seconds where this will stay there. Simultaneously, from E2, which is the upper left chamber, the blood will move to the lower left chamber, which is the left ventricle, which is the event E4. There is a specific time span, say x second, y second, z second, q seconds. Okay. And each one of these times are not unique, it is something like this, something like this. They are, so I have drawn the graph like in axis like this. Okay. So, where your x axis is telling you the time and y axis is telling the intensity of the event. Okay. So, each one of these events are overlapping over one another, and there is one more thing conceptually what you have to understand is this event has a unique frequency. It is something like an alarm clock or something like a timer. So, there is a timer governing these 4 events. There are many other small events which are happening. I am just ignoring them for your understanding. I am just concerned at this point anatomically about all the 4 different chambers which are filling, unfilling, filling, unfilling, filling, unfilling, likewise the event which is going on. So, I am not taking into account the other stuff. Okay. So, there is a timer governing these events. In other words, this timer has a certain frequency. Now, here we are with this basic concept, we are introducing the 2 different systems existing within the cardiac physiology or within the structure, anatomy and physiology of the heart. In terms of the physiology, the process by virtue of which this frequency pattern of filling, unfilling, filling, unfilling, filling, unfilling is being maintained is called the conduction system of the heart. Conduction system of the heart. Conduction system of the heart. And this conduction system is nothing but the frequency which governs it. It is an electrical circuitry which governs this. Right? Second system which is parallelly happening is a mechanical event, which is the filling, expansion, contraction of these four chambers, which is happening in a rhythmic fashion, and that process is called, the next system or the parallel system is called contraction system of the heart. So, it is a conduction system which regulates the motion of the contraction system, right? And this is the controller unit, and this conduction system is an electrical event, whereas this contraction system is an mechanical system. So, it means you can consider the heart as electromechanical system. The electrical system governing the mechanical action of the system. So, this is an electromechanical system in its complete totality. So, now having introduced this concept of conduction system and contraction system, now I will go on to give you the anatomical or physiological analog or the anatomical features which constitute the conduction system of the heart. Now, coming back to the anatomy of the heart, now I am introducing the conduction system. So, conduction system in this whole heart consists of only hardly few percentage of cells, which makes that electrical circuitry, which is a timer circuit within it. So, what is the location of the timer circuit? So, you follow the orange line what I am drawing, and that will kind of give you an idea about how the conduction circuit is being laid out within the anatomical framework of the heart. So, this location, this is called SA node, this is called AV node, I will come to that. This is called bundle of His, and these are called Purkinje fibers, after the name of famous Purkinje, whose name there are Purkinje neurons. Now, SA stands for sino-arterial node, and AV stand for anterior ventral node. So, the way it works is This is a autorhythmic circuit. What do I mean by autorhythmic circuit? It is a automatic circuit which continuously generates an impulse. You must have seen an impulse generator, right? This is a classic case of an impulse generator which is continuously generating an impulse like this. How it is doing, I will come later. What are those electrical features, what made this impulse generator so unique? It is continuously doing like this, it is generating an impulse. So, the impulse gets generated from the AV node, from the SA node. So, out here the impulse is getting generated. So, impulse travels like this. Obviously, this is the shortest distance from SA to AV node, where the AV node is here, whereas this is a longer distance, right. So, the first event E1 happens when the impulse travels from SA node to AV node on the right atrium or on the right upper surface, right. Parallelly, there is an impulse which is traveling from SA node to the AV node via the left upper chamber, which is, if you follow the line what I am drawing, this is it. So, this event is the event 2, what we have named here as E2, right. Then from here, since this impulse has come earlier, this has started to move on this direction. So, here, by the way, there is a slight change I am doing. So, there is a split here. Okay. So, this impulse now already moving like this, spreading here. So, while from the AV node, this impulse is now moving towards the right ventricle or the right lower side, the third event occurs, which is E3. The electrical impulse is now spreading on this side of the heart. Okay. Whereas the last of the events which get initiated from here, where the electrical impulse is, so this is the circuit, like this, like this, like this, like this. Okay. That is where impulse is moving like this, and this one is your E4. So, these 4 events are regulated by this wonderful conduction system, which is so very well laid within the framework of the heart. Is that clear? Now, having said this, how this circuit is autorhythmic? Why it is essential for you to know is because whenever we have talked about electrical activities of the neurons, we have always talked about the cells which are sitting at say minus 70 or minus 80 millivolt, right. Now, think of a situation. I told you, when I taught you that thing, I told you that there are some basic principles which you have to follow, all or none. And if a cell reaches at around minus 40 millivolt, then it is bound to shoot an action potential, right. Now, think of a cell which continuously stayed at a minus 40 millivolt membrane potential. Resting membrane potential is minus 40 millivolt. So, such a cell follows a different paradigm of transmission of signals, and the different paradigm of transmission of signal is what we call as the pacemaker activity of the cell, and the cells which constitute the SA node, AV node, bundle of His and Purkinje fibers are called the pacemaker cells. And pacemaker cells of the heart remains at minus 40 millivolt resting membrane potential, and these cells at minus 40. So, now if I draw the graph like this in terms of millivolt and in terms of time, if this is 0, this is minus, this is plus, and this is minus 20, this is minus 40, this is minus 60, this is minus 80. Similarly, here you have plus 20, plus 40, likewise. Okay. So, what we as of now we have talked about the cells which are standing somewhere here, and then they shoot an action potential like this, right? Now, while teaching that, I told you if a cell sits out here, so I told you that if a cell could reach up to this, then if you could make the cell to go that much positive from minus 70 to minus 40 or minus 80 to minus 40, then at this stage it's all or none if it is an action potential, because it will invariably will shoot an action potential. But what will happen if a cell continuously stayed at this action potential or at this resting membrane potential? Such cells are spontaneously active, and there are different kind of channels which are involved in it, and those cells are shooting spontaneous action potential. So, this is one deviation what I am introducing about cells which do not sit at minus 80 or minus 70. There are cells in nature within our body which are called the pacemaker cells. They sit at an action at a membrane potential of minus 40, and because they sit at minus 40, they shoot spontaneous action potential, and our whole life relies on these interesting pacemaker cells which should spontaneously keep on firing our heart, and the day they stop, the day the end emerges. So, the thing is that if this circuit has a flaw, it has a block or something, the only way is you implant a pacemaker, and that is called implanting of the pacemaker. Okay. So, what is a pacemaker? Pacemaker is nothing but a signal generator, artificial signal generator, which is embedded somewhere out here, which ensures that the impulses are being generated and this circuit is being maintained, because there is a kind of a conduction which happens. There is one more thing which I will close in this class, and I will tell you how this pacemaker activity is integrated with the activity of the contractile system. Okay. So, I will close in here. Thank you for your patient listening.